The overall goal of this procedure is to create adhesive and soluble gradients to study cell migration. This is accomplished by first passivating a surface to prevent nonspecific cell adhesion. The second step is to fabricate stamps from microcontact printing. Next, the passivated surface is patterned with streptavidin lines by microcontact printing. Streptavidin can also be printed as dots using dip pen lithography. Attached to the bottom of a microfluidic device, the modified surface will act as a foundation for an adhesive gradient of biotin RGD. The final step is to load cells onto the surface with the adhesive Q in the presence of a soluble chemoattractant gradient. Ultimately, live cell microscopy is used to show cell migration in response to both adhesive and soluble cues.
The main advantage of this technique over existing methods such as transwell or Boyden chamber assay and a conventional microfluidic setup is that this integrated system of surface pattern microfluidic device allows the observation of cells' responses to spatially controlled adhesive cues, an adhesive gradient, and a soluble gradient at the same time.
Well, this method can help answer key questions in the cell migration field, such as the importance of cell adhesion and crosstalk between chemotaxis receptors, such as receptor tyrosine kinase, G-protein coupled receptors, and adhesion receptors like integrin.
To passivate glass cover slips, first clean the cover slips by submerging them in a rack in 100% ethanol and placing the rack in a sonicating water bath for 30 minutes. Dry the glass cover slips in air. Next, sonicate the glass cover slips for 30 minutes in 1 molar sodium hydroxide. Afterwards, rinse the glass surfaces by carefully dipping the rack in a beaker filled with 1.5 liters of millicube water. Repeat the rinsing process 3 times using fresh millicube water each time. After the third rinse, dry the glass cover slips in an oven at 60 degrees Celsius for about half an hour. Place half of the clean glass cover slips individually in a humid chamber that is made of a 24-well plate partially filled with water. Wells are filled with water and the glass cover slips are laid on top of the wells. For the passivation, a copolymer of polyl-lysine and polyethylene glycol is used in which 20% of PEG molecules are grafted to biotin, abbreviated as PLL-PEG biotin. Drop 15 microliters of PLL-PEG biotin in PBS on each glass cover slip. Take the other half of the remaining clean glass cover slips and carefully sandwich the PEG solution. Leave the cover slips for 1 hour in the humid chamber. After an hour, slide the sandwiched cover slips gently away from each other without scraping the PEG-coated surface, and rinse them with millicube water. The water should slide off easily on the PEG-treated side. If necessary, air dry the glass cover slips on a paper towel with the treated surface facing upward. Store the glass cover slips in a vacuum desiccator until further use. The silicon master required for this procedure is fabricated by photolithography. To cast a PDMS stamp, pour the PDMS elastomer onto the silicon master inside a petri dish to about 1 centimeter in height, and cure the master and PDMS mixture in an oven at 70 degrees Celsius for 1 hour. Subsequently, the PDMS stamp is removed from the silicon master and cut to size. To print protein patterns using the microcontact printing technique, first clean and make the PDMS stamps more hydrophilic by treating the pattern side in a UV and ozone cleaner for 1.5 hours. Immediately after the ozone treatment, place and spread 10 microliters of streptavidin onto the PDMS stamps. If tracks should be visible under the fluorescent microscope, use streptavidin that is conjugated to a fluorophore, such as streptavidin elixifluor 350. Leave for 1 hour in a humid chamber. Remove excess streptavidin from the stamp with tissue paper and air dry the stamp for approximately 1 minute. Lightly press the stamp onto the PLL PEG biotin-coated glass cover slip. If fluorescent streptavidin was used, examine the pattern under an epifluorescence microscope. Protein patterns can also be printed using dip pen lithography. First, mix 1 part of 1 mg per milliliter streptavidin or streptavidin elixifluor 350 with 10 parts of glycerol, and load 5 microliters of the mixture onto the cantilever. Adjust printing speed, contact time of the cantilever on the surface, or vertical distance of the cantilever to the surface to reduce the spot size to approximately 5 micrometers. Begin the printing process as described in the operation manual of the dip pen lithography instrument. Store all printed surfaces in a desiccator. A commercially available microfluidic device is used to create surface gradients. Adhere the streptavidin-coated or patterned glass cover slips onto the sticky side of the device. To ensure that there is no leakage for several hours, seal the edges of the device with a thin layer of warmed Vaseline and with a second layer of a mixture of 1 part Vaseline to 1 part paraffin wax. Fill the channel of the device with 6 microliters of PBS. To create 2 opposing gradients, fill the 2 reservoirs, one with 70 microliters of biotin 4-fluorescein and the other with 70 microliters of biotin conjugated to biotinylated peptide arginine glycine aspartic acid, or RGD. Incubate the samples at room temperature in the dark for 1 hour.
Rinsing biotin RGD and biotin FIT-C off the microfluidic device is the most difficult aspect of this procedure because there's the risk of trapping air bubbles and disturbing the microcontact printed tracks. To ensure success, the biotin and PBS must be removed carefully and slowly.
Remove the biotin solutions and carefully rinse the surface twice with PBS while still attached to the microfluidic device. Mark the direction of the adhesive gradient. Prior to loading fluorescently labeled cells into the microfluidics device, count the cells and divide them into 2 tubes of equal cell numbers. Wash and resuspend 1 tube of cells with media containing the chemoattractant, such as low-glucose DMEM with 10% FBS. Wash and resuspend the other tube of cells in media without the chemoattractant, such as DMEM with 0% FBS. The final concentration of cells in each tube should be 5 × 10⁵ cells per milliliter. Remove all the solutions from the microfluidic device and place it on the pre-warmed microscope stage. Load 70 microliters of cells resuspended in 0% FBS DMEM into 1 reservoir. Load 70 microliters of cells resuspended in 10% FBS DMEM into another reservoir. Loosely place tape over the reservoirs to avoid evaporation. Ensure that the channel of the microfluidic device is in the field of view and cells are in focus. Select image acquisition parameters, such as exposure times and fluorescent filters, image sequence of bright field and fluorescence that capture cells and tracks, as well as time points and recording length. Start the image acquisition program. This video demonstrates a method for live cell imaging of cells that migrate in an environment with competing adhesive and soluble gradients. Adhesive tracks that contain fluorescent streptavidin and biotinylated RGD were created with microcontact printing or dip pen lithography. Successful microcontact printing is indicated by the line profile of the fluorescence intensity across the tracks, where adhesive tracks and anti-fouling regions can clearly be distinguished. Dots printed with dip pen lithography appear rounded, not tear-shaped, indicating a successful printing process. In this example, dots were printed in a line with row and column separation set to 10 to 15 micrometers. This distance is sufficient to prevent dots from merging into each other, but allows the viewing of multiple dots in a single field of view with most microscope settings. A gradient of adhesive cues on the printed track was created with the simple microfluidics device by loading biotin 4-fluorescein and biotin RGD into the opposite sides of a microfluidic chamber. Panel A shows 60 mosaic images taken with a total image length of 3072 micrometers. In Panel B, the fluorescence intensity was measured across the length of the entire mosaic. Linear trend lines were fitted to the fluorescence intensity to indicate the RGD gradient in the channel. After removing the biotin RGD solution, the same chamber can be used to introduce a soluble gradient. Panel A shows the fluorescent intensity of fluorescein dye near the PBS and fluorescein-filled reservoirs and across the channel at T = 0. Panel B shows the fluorescence intensity of the soluble cues after a 16-hour incubation. The comparable results indicate that the gradient was stable for at least 16 hours. HeLa cell migration in a microfluidics chamber with opposing gradients was observed using this method, and a representative movie is shown here. The adhesive gradient was immobilized RGD on streptavidin tracks, and the soluble gradient was 0 to 10% FPS. The images were taken every 15 minutes for a total of 20 hours. The image transition shown is 8 frames per second. This second movie is of a single HeLa cell migrating towards the FBS source. The image transition shown is 8 frames per second. The blue line representing the cell trajectory was obtained via a cell tracking software. This last graphic shows the cell trajectories from the images in the first movie. Cell trajectories that migrated towards the higher concentration of soluble FPS are shown in red. Cell trajectories that migrated towards higher concentrations of adherent RGD are shown in black. These trajectories show that more HeLa cells migrated toward the source of the chemoattractant than towards higher concentrations of adherent RGD.
While attempting this procedure, it's important to remember to avoid evaporation of PBS or media in the microfluidic device during the adhesive gradient preparation and live cell imaging. Use sticky tape if needed to cover all inlets of the microfluidic device.
Following this procedure, other methods like transfection and high-resolution microscopy can be performed in order to answer additional questions, like the biomechanics of the cytoskeleton, actin-binding protein, focal adhesion proteins, and receptors during cell migration.